How does HPV cause cervical cancer? And what are some of the treatment options for cervical cancer? HPV is a sexually transmitted disease which can be transmitted through skin-to-skin or sexual activity. An HPV infection starts when HPV enters the body and into the bloodstream, where it ejects one of its L1 proteins into the bloodstream, where it will be free-floating until it attaches to a proteoglycan on a basement skin cell. Once the L1 protein lands on a proteoglycan, it begins to be enveloped into the basement skin cell, and once fully inside the basement skin cell, it combines with a lysosome, which then lowers the pH surrounding the L1 protein, revealing the L2 protein. The L2 protein is now free-floating within the cell and can enter the nucleus of the cell to release its genome. Once inside the nucleus, the HPV genome begins to export its DNA as RNA into the endoplasmic reticulum, where it can be used to build proteins that will make up the HPV virus cell. Two important proteins are E6 and E7, which will bind to P53 and PRB proteins in the host cell to prevent them from killing the cell. The HPV virus begins to assemble itself with parts made by the host cell, and once there are too many HPV virus cells within the host cell cell explodes, releasing an HPV or many HPV viruses, which will repeat the process. HPV can reproduce within a cell unhindered because two of its proteins, E6 and E7, have bound to PRB and P53 proteins within the cell which usually check to make sure nothing malicious is happening within the cell. With regards to DNA, this is called precancer. Cancerous changes can occur in areas such as the vulva, vagina, penis, anus, mouth, or throat. One treatment option is an angiogenesis inhibitor, known as Avastin, a drug which prevents a signaling cell from signaling the blood vessel to send more blood to a tumor, which would normally allow the tumor to expand. Avastin prevents this and causes the tumor to die. Another treatment option is blocking the E6 and E7 proteins from entering the nucleus. By blocking the pathway that allows the E6 and E7 proteins into the nucleus, they can no longer bind to P53 and PRB and therefore cannot cause precancer. Another option for treating HPV is to use proteoglycans. Proteoglycans could act like antibodies and attach to the active sites on the L1 proteins while they are on the HPV molecule. That way, when the L1 proteins break off from the HPV cell, they cannot bind to a host cell and therefore cannot infect the cell.